This will be an overview of the process of glycolysis and an overview of gluconeogenesis. Glycolysis and gluconeogenesis are two metabolic pathways that the body uses for the production of energy. Glycolysis is an anaerobic pathway that metabolizes one molecule of glucose into two molecules of pyruvate with a net production of ATP energy molecules, NADH molecules, and H+. Gluconeogenesis is a pathway of reactions to synthesize glucose from non-carbohydrate precursors, including pyruvate, lactate, glycerol, and amino acids. Glycolysis stage 1 is the investment stage. The cell makes an investment of energy in the steps of this stage, uses up energy. The goals of this stage are to trap the glucose molecule inside the cell by polarizing it by adding a phosphoryl group. And it also will be destabilized by these same phosphoryl groups in order to prepare it to react for cleavage. And we're also in this stage going to change its symmetry. So in step 1, a glucose is phosphorylated by the enzyme hexokinase into glucose 6-phosphate. This makes the glucose more reactive and makes it polar so that it cannot leave the cell. At this stage, we have lost a phosphate group from ATP and it has become ADP. And the phosphate you can see is on carbon 6. In stage 2 rearranges the glucose 6-phosphate into its isomer, fructose 6-phosphate. And you can see that this is done by an enzyme called phosphoglucose isomerase. So this changes the symmetry to allow for those two similar molecules to be produced from the original molecule. Step 3 adds a phosphoryl group to fructose 6-phosphate to form fructose 1,6-bisphosphate. And I noticed there that they have biphosphate on this diagram, and it's too late to change it, so that should be bisphosphate. It destabilizes the molecule so that it is more likely to undergo cleavage. So you can see now it has two phosphate groups, one on the 1 and one on the 6, hence the name. Glycolysis stage 2. This is the cleavage stage. The goals of stage 2 are the cleavage of fructose 1,6-phosphate to form two identical 3-carbon molecules, and these molecules will continue along the glycolytic pathway. Step 4 of the entire process, fructose 1,6-bisphosphate is cleaved into two 3-carbon molecules that you see there, dihydroacetone phosphate, which is DHAP, and glyceraldehyde 3-phosphate, which we call GAP. And you can see that glyceraldehyde 3-phosphate can continue on through the process of glycolysis. However, dihydroxyacetone phosphate has to be changed into glyceraldehyde 3-phosphate, and that is done with the help of triose phosphate isomerase. So they are isomers of each other, so that enzyme isomerase will change them both to glyceraldehyde 3-phosphate. Glycolysis stage 3, we finally are going to finish the production of pyruvate. Goals in this stage are to destabilize the molecule and produce a molecule that has a high potential to transfer phosphoryl groups. Both GAP molecules that we just produced are transformed into pyruvate in the next 5 steps. So starting with step 6, we're going to transform glyceraldehyde 3-phosphate, GAP, with the help of NAD and orthophosphate, into 1,3-bisphosphoglycerate, which has a higher potential to transfer phosphoryl groups because it has phosphate groups on carbon 1 and carbon 3. You'll notice in this that NADH is formed from NAD, and the enzyme GAP dehydrogenase transfers a hydride group. In step 7, we transform 1,3-bisphosphoglycerate, or 1,3-BPG, into 3-phosphoglycerate by removing the phosphate from carbon 3 and adding it to an ADP to form ATP. And this stage will form energy. In step 8, we destabilize 3-phosphoglycerate by moving the phosphate group closer to the other negatively charged group. So you can see that we have moved that phosphate group up onto the number 2 instead of the number 3 carbon there. And the enzyme is phosphoglycerate mutase, and mutase enzymes change the position of a group. In step 9, we're going to transform that 2-phosphoglycerate into an enol called phosphoenolpyruvate, or PEP. And we're going to do this by dehydration. You see the loss of a water molecule there. And then our last step, step 10, we have the phosphoenolpyruvate, or the PEP, and it has a high potential to give up its phosphate group to ADP to form ATP. So once again, we have produced some energy, and our final molecule there is pyruvate, and it's produced by pyruvate kinase. And this is the whole diagram, and it has an additional step, the changing of pyruvate to lactate at the very bottom with the enzyme lactate dehydrogenase. So this is the net result of glycolysis. And it shows us that starting with one glucose molecule, adding 2 ADP and 2 NAD and 2 orthophosphate, we end up with 2 pyruvate, a total of 2 ATPs, 2 NADH, 2 protons, and 2 molecules of water. Now we will begin our overview of gluconeogenesis. Gluconeogenesis meets the needs of the body for glucose when there is no glycogen reserves available and when the blood glucose level falls below 70. Glucose can be synthesized by non-carbohydrate precursors. There you see them at the bottom: lactate, amino acids, pyruvate, and glycerol. And this process usually takes place in the kidneys, to a smaller extent in the liver, and it has been detected also in the brain and in other areas of the body in emergencies. It is the conversion of pyruvate into glucose following the reverse steps of glycolysis, but bypassing the 3 most exergonic steps of glycolysis, because those would be very endergonic steps in gluconeogenesis. which would mean it would take a lot of energy to do those steps, which is not available. The process uses phosphoenolpyruvate as a starting point and then travels backwards along the glycolytic pathway. Most of the pathway corresponds to the reverse of glycolysis. However, we will have to refer or examine those 3 irreversible steps that have to be bypassed. This is a pretty good comparison diagram showing the differences between glycolysis and gluconeogenesis, at least in the basic steps and their enzymes. So you can just glance down and see where there's a change in the enzyme, which means that there was a change in the pathway because the amount of energy required to get through that step would have been too high. So on the glycolysis side, we move down from glucose to pyruvate, and then gluconeogenesis will start down at the 2-pyruvate. And on this diagram, it doesn't show the 2 steps in between pyruvate and phosphoenolpyruvate, but there's a couple of things that have to happen there. This step takes place within the matrix of the mitochondrion, and what it does is it uses a highly exergonic process of hydrolysis of an ATP molecule in order to drive the process of carboxylating the pyruvate into oxaloacetate. And the enzyme would be pyruvate carboxylate, which is found only in the matrix. And also acetyl-CoA is required, and that's required to bind the carbon dioxide to the biotin binding domain on the enzyme. And then the oxaloacetate and the NADH are reduced using malate dehydrogenase, and it is actually converted temporarily. Oxaloacetate is converted to malate in order for it to be transferred across the membrane of the mitochondria into the cytoplasm, and then it's transferred back to oxaloacetate. And then finally, oxaloacetate is phosphorylated into phosphoenolpyruvate using the energy of the decarboxylation and the release of the CO2. And so now we are at stage 2 at the phosphoenolpyruvate. From there, you can see that the enzymes are the same. So the process from this point is simply a reverse of the steps in glycolysis using the same enzymes. These steps have Gibbs free energy value that's close to 0, and so they happen very easily. They don't require any extra energy. In step number 7, allylase combines the glyceraldehyde and the DHAP to form the fructose 1,6-bisphosphate. And then you can see that is a new enzyme on the right-hand side than was on the glycolysis side. So the conversion of fructose 1,6-bisphosphate to fructose 6-phosphate requires a different pathway than glycolysis. It would require the input of too much energy to use the same pathway. So the enzyme fructose 1,6-bisphosphatase hydrolyzes an ester bond on carbon 1, which is an exergonic energy-releasing bond, and the process moves forward. And then fructose 6-phosphate can be readily transformed in the same way that it was transformed in the other direction to glucose 6-phosphate using the same enzyme as glycolysis. And then finally in step 10, you can see that there is a different enzyme there. Oftentimes glucose 6-phosphate ends the process, and the reason for this is because it can be easily stored. Glucose 6-phosphate is the one that cannot escape the cell because it's phosphorylated. So it can't escape the cell. So as soon as you change it into glucose by removing the phosphate with the glucose 6-phosphate, it can escape the cell. So if you are wanting to store it, you wouldn't change it, you would keep it that way, and then it can be transformed easily into glycogen and be stored that way. In the liver or kidney, if they are needing to regulate the blood glucose, then they would transform that glucose 6-phosphate into a glucose. And step 10 is definitely a different type of step. The glucose 6-phosphate must be transported into the endoplasmic reticulum, and there's 5 different specialized proteins that are part of that job of getting it into the endoplasmic reticulum and being an enzyme on the inside of the membrane. And then there's also a calcium binding stabilizing protein, and then there is a transfer protein, another transfer protein. So there are several different proteins that are needed just to change that glucose 6-phosphate into glucose. And so that is our summary of gluconeogenesis. The enzyme autolysis, we saw it earlier in glycolysis and in gluconeogenesis. It was responsible for transforming that fructose 1,6-bisphosphate into the glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. So the 6-carbon sugar into the 3-carbon sugars. It also went the other way in gluconeogenesis and catalyzed the transformation of the 2 3-carbon sugars into the 6-carbon fructose 1,6-bisphosphate. High amounts are found in the muscle tissue. That makes sense since there's a lot of energy need in muscle tissue and a lot of breakdown of glucose. If higher than normal amounts are found in the blood, it may indicate damage to the skeletal muscles or to the heart, or it may indicate that there's a muscle disease, or even it's been indicated in liver disease. Autolysis has isozymes. Vertebrates have been shown to have 3 autolysis isozymes called A, B, and C. These isozymes have different electrophoretic and catalytic properties. They are found on different chromosomes, the genes for these isozymes. A is found on 16, B is found on 9, and C is found on 17. They're found in different tissues. Autolysis A is found to be expressed in red blood cells and muscle and brain. Some studies show the brain. Autolysis B is expressed in liver and kidney and intestines, and autolysis C is expressed especially in the brain. In addition to the metabolism and glycolysis that we've been looking at, autolysis has been associated with non-enzymatic functions through protein interactions, epigenetic modifications. It's been validated as a prognostic marker for human cancers, and aberrant expression has been observed in several human diseases and cancers. To finish up this presentation, I'd like to look at this autolysis B and fructose study. It turns out there's an autosomal recessive condition called hereditary fructose intolerance that results in a deficiency of autolysis B. And this deficiency leads to liver and kidney dysfunction immediately following fructose consumption. That's interesting because the autolysis B seems to be working for regular glycolytic pathway and for the regular metabolism, but fructose is especially dangerous. And for infants, it can lead to death. And the rate may be as high as 1 in 20,000 deaths. These are infants who just begin to eat adult food or even children's food that is containing fructose. So it helps you to realize that we may know one function of one enzyme, but the way that our body is designed, it seems likely that one enzyme has many different jobs, and we should never be sure that we know everything about a certain enzyme. We should continue to research. Thank you.